So it is my pleasure to introduce Davide Cozzi, who is a PhD student at the University of Milano Bicocca in our group. Davide will describe some of the research problems and of the recent advancement in data structures in pangenomics. As you can imagine, we have a huge problem in pangenomics, that is the data is very large and it is difficult to query and it is difficult to extract efficient information out of that data. And so Davide will show you what techniques must be used in order to access and store those data efficiently. So thanks again, Davide, for giving this talk, and please go.
Thank you, Professor Della Vedova. Good morning, I'm Davide Cozzi. As told before, I'm a PhD student here at University of Milano Bicocca, and today I will talk about indexing pangenomic haplotype data using the positional Barui transform. This will be the schedule for this talk. First of all, I will describe what is the positional Barui transform, the PBWT. And if you remember the talk by Simone Rubinacci, it was also introduced at some point in his slides. Then we will see a sort of overview of some computational problems that are solved using this particular data structure. We will see new research topics on improvement the PBWT. We will see some extension, and more in detail, we will understand what I have done in my first 2 years of PhD, because I will describe my MOO PBWT. And then we will see some examples of real-world uses of this data structure. You have seen this kind of pipeline many times in these first 3 days. We can think about our input data as a set of haplotype strings, but we want just to focus exactly on the SNPs on variation site in order to not study and not store in memory entire sequences, but just the minimum differences between them in order to have a feasible computational data to analyze. So we are computer scientists, and we need to understand what we are trying to index and store. In our case, we are looking for binary metrics, where each row represents an haplotype, and each column biallelic site. So in this first part of the presentation, we will just focus on binary metrics with an alphabet of 0 and 1, but we will see that there are some improvements in extending also the alphabet size. Then we have 2 main classes of problems. The first one involves find matching, find similarities between the haplotypes inside the reference panel. But we can also consider, as you see in this slide, an external query, so a query haplotype, that we want to compare it against our reference panel. On blue, I have just highlighted some of the possible matches that are interesting in analyzing this kind of binary metrics, and they are called set maximal and set matches, but we will describe more in details later. So I know this will be how can I say, a more algorithmic talk instead of all the other ones in this first 3 days, but I think it should be interesting to also understand how data structure works, and maybe someone will find this kind of methods interesting and will start some new research project or include in their current research project. So we are studying haplotype as binary W-long sequences. A panel of haplotypes is just a set of these haplotype sequences. And we are interested in external haplotype that has the same length of any other haplotype in our reference panel. To give a sort of abstraction of what is a match, we can define a set match as a triple ABE, where A is a set of haplotypes, and with this triple we represent a match between these two columns, B and E. Then we can define more strictly a set maximal exact match. In this case, it's with an external query, but we can think about Z as another haplotype in our reference panel. And this kind of set match, as you can see in these three dots in the last block, this match cannot be extended neither to the right or to the left, and we cannot include any other haplotype without losing this property of having a match. So, positional Barrow-Wheeler transform. It was introduced by Richard Darwin in 2014 as an interesting data structure able to store and query this kind of binary metrics, and is strongly based, as you can understand by the name, on the ideas of the Barrow-Wheeler transform. Also, Richard Darwin just focused on binary metrics. How does it work? Consider that as the classical Barrow-Wheeler transform, we want to permute the input matrix in order to allow efficient queries. So it works using this colexicographical ordering. So at each column, we reorder from the right to the left. This is colexicographical, in order to make similar rows consecutive in our permutation. Let's see an example, because it's really hard to understand without an example. We are trying to index, for example, column 5. So we sort all the rows up to the previous column, in this case the column 4, and we store the ordering of this of the haplotypes in the so-called prefix array, because the PBWT is just a set of 2 arrays per column, de facto. Then we can store the common length between 2 consecutive rows in this reordering, the common length of just the starting column, such as in the example. And as you can see, thanks to this kind of reordering, similar rows are consecutive, so they share a long common suffix. This property is also useful because if we take the column 5 permuted thanks to this reordering, obviously this is an example, but just imagine real data such as the ones from the 1000 Genome Project, where we have very few ones. We obtain that similar rows are consecutive up to a certain column, so we can likely expect that also the next column will, 2 rows will share the same bit. So this produces the so-called runs. And this is the key point of how to make efficient in memory the PBWT. In this example, we can see that, for example, we have a run of 2s, then a single 1, a single 0, a run of 4 1s, and a run of a lot of 0s. We can store just the number of runs and the bit represented in each run, and obviously the length of the run. Okay, again, we are computer scientists. What does it mean, efficient? In this case, efficient means that we can compute all the set of prefix arrays, all the set of divergence arrays, and also each PBWT column And if we consider all the set of PWT column, we obtain the PWT matrix in a time that is linear in the size of the matrix. And it's not something so, how can I say, easy to obtain. I mean, before 2014, it was not so simple to just produce an index in linear time that is so efficient in allowing queries. Okay, we have indexed our panel, but what we can do with this index? Richard Tarpin proposed three main problems that can be solved. First, find all matches within the reference panel longer than a minimum length. So we are talking about all versus all haplotype matching with a threshold on the length. Then we can find all the set maximal matches, again, within the reference panel. And we will see very soon how many of these matches can we get from also a toy example. So just imagine that how efficient should be this data structure to allow this kind of solutions on real world data. And then we also can find set maximal exact matches from an external query to the reference panel. This is the entire set of set maximal matches within this toy example panel. As you can see, they are a lot. So just imagine how many they can be on, for example, 1000 Genome Project data, chromosome 1, where we have about 5,000 rows and 6 million columns. On the other hand, if the matching is guided by an external haplotype, the number of matches obviously decrease. And as you can see in this example, we have a match starting from column 0 to column 5, shared with 4 rows in our reference panel. Then we have an overlapping match with just 1 row, another overlapping match with just 1 row, and finally other 3 rows that share a match with the query. As said before, we say efficient, but how much these algorithms are efficient? For the L-long matches, the Richard Darwin proposed an algorithm that runs on, okay, this is the real complexity, but just focus on the HW sides. So we can say this algorithm runs in the sides of linearly in the sides of the matrix. The same amount of time is required to compute the all set maximal matches within the reference panel. On the other hand, computing the set maximal exact matches with an external haplotype is faster because it's just linear on the sides of the external haplotype. So it's big O of W. We can ignore the number of matches because they are. We can approximate this complexity bound to big O of W. But we cannot just focus on time. We also need to take into account space bounds. And this is the key point of the PWT, because each type of query that consider only the reference panel, so for example, the first 2 problems, can just run while we are building the index. So we need to just store the information for a single column and output also the matches. So we have a very light data structure in terms of memory usage. On the other hand, if we want to compare an external haplotype, in this case, we need to store a lot of additional arrays that takes a lot of memory. We are talking about something that is linear in the size of the matrix, but in reality, this is linear, okay, but a lot of bytes. In fact, if you do some math, we can obtain 13 HW bytes. And if you remember the numbers that I told you before regarding the chromosome 1 panel from the 1000 Genome Project, we obtain something that is like 400 gigabytes of RAM. So it's not something that everyone can provide. Just some other consideration. The first one is already done because it was about storing just the information for one column per time. The second one is really interesting because Richard Darwin also developed an efficient algorithm to perform external queries that just merge the queries inside the reference panel and perform the set maximal exact matches finding within this new panel. This algorithm is not well described because it's not described in the paper. I honestly don't know why. But it's really hard to extend to solve other kind of problems. So this is the reason of the development of the MuPWT, and we will see how later. We can also solve other kind of problems. For example, set maximal exact matches with a coverage, because I mean, in the example seen before, we have some SMEM with just one line in the reference panel. Is this enough to provide some meaningful result? I'm a computer science, not a biologist, so I like to provide also some algorithm that put more strong results. So provide a coverage of a minimum k lines in the reference panel. And here we can see an example of k equal 3, and obviously all the matches with just one line disappear, and we have smaller matches in terms of column, but obviously bigger in terms of rows. Then we have another very interesting problem that is called the maximal blocks problem finding problem. And in this case, block is just like a set maximal exact match, but with some less restrictions. In fact, we can output a block every time we are losing a row. So let's look at an example because probably it's easier. If you look, for example, to the yellow rectangle, this is a block, but this is not a set maximal exact match because if we are computing the SMEM, we can extend the 0 line and the 2 line because we have one in column 5 on the other hand, and obtain, for example, this set of SMEMs. But if you are looking for blocks, we can just output also this internal match. Blocks should be really interesting to understand similarities inside of our reference panel, and also they are used to build, for example, pangenome graph. We will see something later. Last problem that can be solved using the PBWT. This is the minimal position substring cover, and this is a problem I worked on during the last year. In this case, we want to cover an external query using a set of substrings that are completely covered in our reference panel. So each position of the pattern must be covered, and obviously we are interested in minimizing the size of this set of substrings. This is an example. Again, we have a strong correlation between this problem and the set maximal exact matches finding problem. We have, for example, a coverage that is exactly the first SMEM in the left. Then we are not interested in overlaps, so we can just skip the first 3 columns of this match and just focus on the last 5, and we continue until scanning the entire pattern. We have some variants of this problem in literature. We want, for example, the leftmost MPSC that consider the MPSC set at the start as least as every other possible MPSC, or rightmost is the opposite, so ends at least as late as any other. Or again, we can impose a sort of coverage requiring to have at least k rows in our panel that provide the coverage. So this is an overview of the main problems, but the development on the PWT just doesn't stop with Richard Darpin. We have a lot of variants, a lot of extension proposed in literature in the last more or less 10 years. As introduced before, we can have the multi-allelic PWT, so we are not focused on binary alphabet, but an arbitrary ones. Then we have the so-called dynamic PWT that does not rely on arrays for storing the prefix and divergence arrays, but they become linked list in order to enable the insertion of new haplotype. And this can be very useful in a real-world scenario. Then we have some improvements in terms of computational time. For example, in this case, solve the L-long matches. And then we have something that is more related to compression of the index of TWT, because data are becoming larger every day. If we think about UK Biobank data, we have panels with hundreds of thousands of rows and millions of columns. So we need to compress more also this space-efficient data structure. And also we can focus on computing in parallel, because if you remember the description of the PWT, we need to proceed column by column, but there are some projects related to how we can build, for example, starting from different point in the panel and then trying to correct every time it's not the correct index. Now a sort of sponsor for our laboratory, because this is the wild PWT developed by another PhD student on our lab. And in this case, the project is focused on finding maximal blocks in panel with errors or with missing data that are represented here in the example with a star. And the star is a sort of wildcard. It can be anything. So as you can imagine, this problem is exponential in time. And using the PWT, it remains exponential, because in the worst case, it remains exponential. But it can easily run also on real-world panels with an error rate of 1, 2, or 5%. And yes, this algorithm should be used also to eventually correct the reference panel. So, just an overview of my main project. This is the MoPWT. If you are used to the last, well, not last, because the R index PWT is developed, I mean, I think 10 years ago, but okay. We tried to use this run to index the PWT, and we developed, in collaboration with all the guys you see in the first point of this list, a sort of R index for the PWT. So we just wanted to perform queries, not more in linear time of the, in space, not more in linear space, but in sublinear space. So focusing on the number of runs. And the first problem we solved was the set maximal exact matches with an external query. and we want to outperform these 13 HW bytes. That was the trade-off, strong trade-off on using the Darpin's algorithm. This data structure is strongly based on such data structure, so it's really light, but really fast to query. And we also extended the use to solve the k-set maximal matches and the MPSC problem. I won't go into details because we don't have enough time, but just think about that each of these components that are the PWT are stored in a space that is proportional to the number of runs. I will skip this example because it's really too hard to describe. But let's just see the results. These blue dots represent the PBWT developed by Darwin. And as you can see, for the chromosome 1 of the 1000 Genome Project data, well, chromosome 1, but also chromosome 2, that is bigger, we are talking about 400 gigabytes of RAM for performing the set maximal exact matches. On the other hand, PBWT, that is the orange one, just require more or less 5-6 gigabytes of memory. Without any, yes, we are a little slower, but not so much. And also the index file is very light in memory if we consider that the BCR file is the binary input. We have 2 gigabytes of index compared for 0.5 gigabytes of index of input file, so we are okay. And we can also scale on UK biobank data. These are the results for the chromosome 20. And as you can see, these panels are really sparse, and more the panel is sparse and more runs and less runs it has. So this is the best scenario for us, but we are lighter than our index is lighter than the input size. So it's really, really an interesting result. And to conclude this talk, I want you to present some projects that uses the PBWT to solve real-world problem. First of all, punch blocks, because it's developed in our lab and it will be explained in the next talk. That uses the PBWT to compute the set maximal blocks, starting from multiple sequence alignment, and then uses these blocks to build a pangenome graph. Then we have PRIMS2 and Impute5 that are already described by Simone Rubinacci in the first talk of today. Then we have pSmoother that is used to correct errors in order to provide better panels for downstream analysis, but you can also perform IBD detection, again, as told by Simone Rubinacci in the first talk. And then nowadays I'm focusing on this problem, that is finding haplotype pairs in the reference panel that can resolve an input genotype. So we have also heterozygote sites in the genotype and want to find the best pairs of haplotype that resolve this input. So this is all for today. Probably I have been too fast, but I think it's okay. We have time for questions if you have something. And thank you so much for your attention.
Thanks, Davide. We're waiting to see if there are some questions from the audience. Otherwise, my first question is, while the standard Peirce-Wheeler transform is quite well known and is very widespread in bioinformatics, the positional variant is still not as widely used. do you have any idea or any opinion of why it's the case?
Well, probably we have many problems that are related to linear reference. So we are looking for alignments, we are looking for maximal matches finding problem on a single sequence file. So a single FASTA, a single sequence. And obviously we have many problems on this kind of input instead of haplotype panels. And on the other hand, we are obtaining many more data just in the last few years. And the PWT was developed just 10 years ago. So probably we need some time to make this data structure also suitable for biologists and less theoreticians, people to use it. I don't know, honestly. This is my suspect.
Thank you very much. And then another question is, how do you think that those techniques can be used to index pangenome graphs?
Well, pangenome graph, this is probably a simpler representation of a pangenome graph because we are skipping the common parts of. I mean, if you think about a pangenome graph, we have, for example, a node that is shared between, for example, just a simple example, all the haplotypes, and this kind of information is completely missed in our haplotype panel. While on the other hand, if we have a bubble, we are just storing more or less the same information. But I don't know, probably a sort of mix between storing a reference and the PPWT of a panel can allow some similar query, but probably the graph is the best solution in this in common tasks like alignments or exact matching.
Okay, thank you very much. There are no more questions from the audience, so we thank you again for giving this talk.
Thanks.
And we are going to resume